Hello everybody. In this video we will discuss viruses, its morphology, chemical nature, reproduction, and also a few words about microscopic methods of microbiology research. Let's start. So virology, it's a science about viruses and disease that they cause. Viruses are tiny infection agents that can only replicate inside the living cell of organism. So that means that they can't replicate by themselves. They use host cell for replication, for reproduction, and it's very important to understand viral morphology and viral nature to protect viral infection and control viral infection. The father of virology is Dmitry Ivanovich Ivanovsky, who was the first who told about tobacco mosaic virus. And what is viruses? Its general properties. Viruses are too small. They are very simple. Acellular. They don't have cellular structure. They are too small. They are ultra microscopic. They contain either RNA or DNA. They are always obligate intracellular parasites. They need a host cell to reproduce because they depend on the host cell. They reproduce inside living cell. They can't reproduce by themselves. They are not sensitive for antibiotics. They can cause different infection in human body, in animals and plants. They are much smaller than bacteria. They are submicroscopic. While most viruses range in the size from 5 to 300 nanometers, in recent years a number of giant viruses, including mama virus and panda virus with a diameter of 0.4 micrometers, have been identified. So look at this slide. Here we can see eukaryotic cell, erythrocyte, and the comparison with bacteria cell. Bacteria is much smaller than erythrocyte, but look here. Here the biggest virus, smallpox virus, and in comparison with bacterial size, we can see that virus is much more smaller. They are one of the smallest microorganisms. Viruses can exist in two shapes, actually in three shapes, but main shapes are helical viruses. Helical viruses have an elongated tube-like structure with the capsomeres arranged helically around the coiled genome. And ixoedral viruses or cubic viruses have a spherical shape with the ixoedral symmetry consisting of 20 triangular faces. But some viruses can have the shape which include helical part and ixoedral part, and that viruses are called complex viruses. Look at this picture here. Here we can see cubical, hexaedrical viruses and helical viruses. Here, cubical, like ixoedral. The example is parvoviride, adenoviride, herpes viride. Viral morphology. Virus has a very simple morphology. They have one main structure, nucleic acid, which can be RNA or DNA, not both, only RNA or DNA. And their nucleic acid is surrounded by a protein coat, which is known as a capsid. protein coat which is known as a capsid. The capsid is composed of smaller protein components referred to as capsomers. The capsid and genome combination is called the nucleocapsid. Viruses can also have an additional membranous layer that surrounds the nucleocapsid and called an envelope. Viruses which have envelope, they are called enveloped viruses. The envelope is actually acquired from the nuclear or plasma membrane of the infected host cell and then modified with the viral proteins called peplomeres. Some viruses contain viral enzymes that are necessary for infection of a host cell and coded for the viral genome. Okay, look at this picture. Here we can see viral morphology. Each virus might have nucleic acid. Nucleic acid can be RNA or DNA, as I said previously, and it's obligatory for viral nucleic acid to be coated with a capsid. Capsid, it's a protein structure which consists of protein particles, which is called capsomers. Capsomers. And if viruses consist of only two parts, nucleic acid and capsid, we call them naked viruses, non-enveloped. But some viruses, they have additional shell envelope, which is made from plasma membrane or nucleic membrane of the host cell, and viruses which have this structure is called enveloped viruses. Okay, here we can see non-enveloped and enveloped viruses. Virion is a complete extracellular virus particle. And also here we can see the difference between cubic hexahedral symmetry and helical symmetry. Helical, it's like in chain, and hexahedral like a cubic. And here we can see structure of enveloped viruses. Let's repeat, enveloped viruses, they must have a nucleic acid covered by the capsid, and then they have additional shell, which is called envelope. Envelope is consist of proteins, lipids, and glycoproteins. Viral genome, it can be RNA or DNA. As there might be single-stranded, mixed-stranded, double-stranded, or double-stranded. RNA can be positive or negative. RNA can be non-segmented or linear, or it can be segmented in a few particles. DNA can be linear or circular. Classification of viruses is complicated because of difference and a lot of information about viruses. The International Committee on Taxonomy of Viruses developed the current classification system and wrote guidelines that put a greater weight on the certain virus properties to maintain family uniformity. All viruses are classified into families on the basis of presence of or absence of envelope, characteristic of its their nucleic acid, DNA or RNA. It's a main and it's a key features, type of DNA, type of nucleic acid, and strategy of genome replication. Okay, viruses, they belong to the kingdom vira and phylum vira, and then they have subdivision on deoxyvira, viruses which contain DNA, and ribovira, viruses which contain RNA. And then they divide to the classes, order, family, subfamily, genus, and species. But usually we not use species. We just use genus for naming viruses. Okay. As I said previously, viruses, they need host cell for replication. They can't reproduce themselves without host cell. And let's talk about how do they replicate, how do they multiplicate inside the cell. This process is not easy and it takes a few steps. Attachment, penetration, synthesis, assembly, and release. Let's talk about each of the steps. The first step is attachment. Attachment is it's a main step because because in result virus will enter to the cell and cause the infection or not if it can't attach. Outside of their host cell, viruses are inert or metabolically inactive. The attachment itself is highly specific between molecules on the outside of the virus and the receptors of the host cell surface. This accounts for the specificity of viruses to only infect particular cell types or particular hosts. Um, so on the surface of the host cell, there are some receptors, and each virus has its own receptor. It can interact only with one type of receptors, and and this is explain why viruses can damage one type of tissue. For example, we have respiratory tropic viruses, we have enterotropic viruses, we have neurotropic viruses, and hepatotropic viruses. It is because viruses must interact with the receptors on the surface of the cell. And do you know that each cell, each tissues, they have its own receptors, and viruses can interact with one type of receptors. That's why this process is very special, and it explains the uh, ability of the viruses to damage one type of tissue, one type of system. But sometimes, viruses can have a poly uh, polytropic ability, so they can damage few types of tissue. It is explained because they have ability to attach receptors that presence not only on one tissue, but one type of receptor can be presented on different tissues. That's why these viruses can damage all of them. So this is the most important step of the viral reproduction. If virus can't find this receptor, the infection will not occur. So attachment, and then second step, penetration. After attachment, viruses they cause they induce penetration. Naked viruses usually use uh, penetration by endocytosis. They made host cell to engulf them. But enveloped viruses, they usually use way of fusion of the envelope with the cell membrane. And then after fusion, virus destroyed this membrane and goes to the cell and goes inside the cell. Next is deproteinization. After entering after entering viral, nucleic acid must become free, so capsid must be destroyed, and then viral uh, viral nucleic acid goes to the process of genome replication and protein synthesis, because you know that to create a new viral particle we need nucleic acid and we need proteins for building, for capsid building. Some viruses, they have specific enzyme which initiate the process of genome replication, but other viruses use enzymes of the host cell. Protein production is tightly controlled to ensure that components are made at the right time in viral development. So, viruses use host cell ribosome to synthesize their proteins. The next step is assembly morphogenesis, viral uh, when viral nucleic acid is associated with proteins and capsid covered this nucleic acid. They are ready to go on, and they are ready to release. Release means that they leave the cell. A majority of viruses leave the cell by lysis, so they kill host cell and go out to find a new cell. But other viruses use budding. This is because they need to create envelope, and in the process of budding, they modify cell membrane, and then they coat with the cell membrane and create their um, envelope from cell membrane. The cell membrane is modified by the insertion of the viral protein with the nucleocapsid portion of through this modified portion of the membrane, allowing it to acquire an envelope. Look at this next picture. Here we can see how virus can modify host cell membrane with viral proteins. They prepare cell membrane to become viral envelope, and then in the process of budding, uh, virus just take part of the host cell membrane and create uh, and, and create its envelope. So this is a very important information. You should understand that envelope of the enveloped viruses, it's a cell membrane which was modified by viral proteins. That's why it can fuse its envelope in the process of penetrating, because envelope of the enveloped viruses is um, have a similarity with the cell membrane, because it's a part of cell membrane but modified it with viral proteins. And host cell reaction. What will cell feel in this process? What will be result after viral reproduction of the cell? Of course, uh, viruses can kill the cell. They reproduce, they multiply inside the cell, release and kill the cell. It's a cytosol infection. And also they can cause apoptosis. Viruses not kill the cell, but cell decide to die, decide to turn off the mechanism of suicide. And also non-cytocidal infection means that viruses also not destroy the host cell, but immune system, immunological reaction kill this cell. So one of the results of this interaction between viruses uh, and uh, host cell is cell death because of viral action, because of apoptosis, and or because of immune reaction, immunological reaction. And also, viruses can cause latent infection. That means in the process of latent infection, viral genome integrate to the genome of the host cell and not destroy and not cause any destroying, but create the storage of the viruses. And that's why we can have chronic infection, chronic viral infection, latent viral infection, viral carriage like that. And also, I think you have heard that some viruses are associated with cancerogenesis. This is because of transformation of the genome of the host cell. Uh, Viral oncogenesis is associated with hyperactivation of proto-oncogene. What is proto-oncogene? It's a gene which are normally present in the host cell, in the animal cell, and their function is regulation of the cell growth in the broadest sense. Proto-oncogenes are potentially contributors to tumor development that have to be activated before they can actually have such effects. So proto-oncogen it's a genes which are normally present in the animal cell. They needs for growing of the cell. But if they will be hyperactivated, this growth will be over that it must be overgrowing. And this genes can be activated because of several different mechanisms, and one of them is activation by oncoviruses. Oncoviruses promoter may induce overexpression of the proto-oncogene, and this is resulting in a tumor. Which viruses associated with oncogenesis? Adenoviruses, herpes viruses, hepatoviruses, papillomaviruses, and other. And also, what is latent infection? Latent infection, it's a type of infection when viruses integrate their genome into the genome of the host cell, but no new viral particles are produced. So virus goes into the host cell and not produce any new particles, not multiplication, but integration to the genome of the host cell. The cell is accordingly not damaged, and the microorganism does not manifest disease. So usually in this case, we have no any symptoms of the disease, but virus creates the storage of itself in the host cell. This form of infection is found, for instance, with the adenoviruses group and particle of the herpes viruses, which can remain latent for long periods in the human body. When viruses keep themselves inside the genome, they are not sensitive for the action of immune system, for the action of antiviral drugs, and this is like a storage. They keep themselves visible for immune system. Uh, the Best example is herpes virus. You know that herpes viral infection, it can be like a chronic infection with repeating, and that's why when viruses enter to the human cell, they cause they multiplied in the mucous tissue and destroy mucous tissue. That's why we have manifestation. But then this viruses goes to the bloodstream and then goes to the neural cell and creates a storage in the neural cell. Inside the neural genome, viruses are not sensitive for the action of immune system and antiviral drug. That's why we can't treat this infection. We can't eliminate these viruses from the body. And when immune system became lower, become weaker, or in the case of stresses, viruses disintegrate from the neural cell and goes to the bloodstream and goes to the mucous and cause manifestation. And then they repeat their circle, goes to the bloodstream and to the neural cell. This is a latent mutation. Okay, about acellular microorganisms, you know that there are not only viruses but also viroids, prions, viroids, small infection agents that cause disease of plants, and viroids they usually uh, have only nucleic acid without a protein coat. Um, and prions, it's a abnormal proteins which can infect, which can cause the abnormal of another proteins in the tissue. Bacteriophage, also cellular microorganism. And they are very similar to the viruses, but viruses, they replicate inside the eukaryotic cell, and bacteriophage, they multiply, they need bacterial cell for multiplying for reproduction. We can call them bacteriophage or just phage. And it consists of nucleic acid, usual DNA, and then it's covered by the protein cords in cubic symmetry, and then it has a protein tail like helical symmetry, and also it has the fibers for interacting with bacterial cell. There are two types of phage, viral and temperate. Viral phage interacts with bacterial cell and causes its infection and kills the bacterial cell. But temperate phage, they have two options. They can interact with bacteria by two ways. Option one is to mimic a virulent phage, following the 5 steps of replication and leaving the host cell at the end, referred to the lytic cycle. So, one way of interaction with temperate phage is to attach the bacterial cell, inject its nucleic acid, replicate uh, uh, inside the bacterial cell, create a new bacteriophage particles, and kill the cell and release it. But two-way is like a latent infection when bacterial phage can fuse, can integrate its genome to the genome of bacteria without destroying bacteria. And this process is known as lysogeny or the lysogenic cycle. In spite of lytic cycle, when bacteria will die after bacteriophage reproduction. Uh, A phage employing lysogeny still undergoes the first two steps of a typical replication cycle, attachment and penetration. Once the viral DNA has been inserted into the cell, integrated with the host cell DNA, forming a prophage. This process is very similar to the latent infection of the viruses of the eukaryotic cell. The infected bacterium, the infected bacterium is referred to as a lysogen or lysogenic bacterium. In this state, the virus enjoys a stable relationship with its host, where it does not interfere with the host cell metabolism or reproduction. The host cell enjoys immunity from bacteriophage. Look at this picture. This is lytic cycle. Bacteriophage inject its DNA to the host cell and then start to replicate its genome and synthesize its code, its protein capsid, and then assembly and releasing by lysin. This is the lytic cycle means bacteria will die after phage reproduction. And this way is usual for virulent phage and temperate phage. Lysogenic cycle means that after injection, phage DNA not goes to the process of reproduction, but it goes to the bacterial chromosome and integrate to the bacterial chromosome and became a prophage. When bacterial multiplicate, it uh, it gives this part for all of their uh, generation. All daughters and sons of this bacteria will have this prophage because bacteria multiplied by binary fusion, because bacteria just make a copy of its genome and also of course it will copy the part with the phage. And why we call this process lysogenic? This is because of possibility of going to the lytic cycle. Each bacteria who has phage its inside its genome, it's possible can goes to the lytic cycle. That's why we call lysogenic cycle. Uh, Lysogens can experience a benefit from lysogeny as well. Since it can result in lysogenic conversation, a situation where the development of prophage leads to a change in the host phenotype. One of the best examples of this is for Caninobacterium diphtheria, the causative agent of diphtheria. The diphtheria toxin that cause the disease is encoded with the phage genome, so only Caninobacterium diphtheria lysogens cause diphtheria. So benefit of bacteriophage is that they can bring some genes from donor bacteria to the recipient when they integrate to the recipient genome. Few words about prions. Prions are different from both viruses and viroids in that they consist of protein and not nucleic acid. Prions are an abnormal isoform of the proteins. It's a post-translationally modified normal host form, which is insoluble in detergents and resistant to protease and nucleases. Proteins are extremely resistant to inactivation by ultraviolet light and ionizing radiation and unusually resistant to alcohol formalin boiling. When a prion enters a healthy organism, it induces exine properly folded proteins to convert into the disease-associated prion form. The prion acts as a template to guide the misfolding of more proteins into prion form. These newly formed prions can then go on to convert more proteins themselves. This triggers chain reactions that produce large amount of the prion form. Here we can see normal prion and pathogenic prion. So the pathogenic prion is abnormally form of the proteins, and then they can um, change normal proteins for the form of primes. So this is a chain reaction. Browns disease is a group of human and animal fatal neurodegenerative disease. Histologically, all Browns disease are characterized by spongiform vascularization in the central neural system, the accumulation of unique protein in cytoplasmic vesicles or neurons, the formation of extracellular amyloid plaque composed of prions. The incubation period of slow prion disease is long, many years. Chronic progressive pathology without remission or recovery, lethal outcome. It's one of the main characteristics of prion disease. And the last question is method of microscopy, which can be used in microbiology research. The microscopic examination of clinical specimen is used to detect bacterial cell, fungal elements, parasites, and viral inclusion present in infected cell. So to research morphology, to find bacteria, to find viruses, to find fungi in the clinical material. Okay, we have principally two methods, electron and light microscopy. Light microscopy means basic light, immersion, dark field, phase contrast, and fluorescence. Usual light microscopy, it's a with using biological light microscope. The effect principle of method is uses of visible light rays, see the cause of physics to understand this process. We not usually use this method of microscopy because our objects are too small, and we need to use and we need more um, uh, we need another method to see our objects. That's why we usually use light microscopy with immersion objective. And the main effect is in coefficient of refraction of immersion oil, which is placed between glass slide and objective lens. You need just drop one you just need to put one drop of oil on the slide, and this oil will increase the refraction. Coefficient of refraction of glass and oil. And this method is most usually used in the microbiology to to um, to research morphology of bacteria and fungi. But we can't see viruses by this method. Okay, look at this slide. Here you can see how does it work. So this is usual without immersion oil, and refracted light rays load the lens like that. And after immersion, we can see that all rays entered to the lens and refraction became bigger. This is a usual picture of immersion light microscopy. Uh, Next method is dark field microscopy. This is also light microscopy but with using specific dark field condenser. And in this case, only light rays scattered from the specimen object reach the objective lens. And this process is this microscopy can be usually used for bacteria can who weakly uh, stain it, poorly stain it. But you know that for immersion microscopy, it is necessary to stain the object. And if bacteria are not sensitive for the staining, we can use dark field microscopy because it does not. This method does not need staining. Look at this picture. Here we can see uh, how difference between usual light microscopy and dark field microscopy. In dark field microscopy, we have specific iris diaphragm and specific condenser, dark field condenser. That's why look the difference between rays which goes to the objective. Here, light waves enable to enter an objective, and there they go to the objective because of action of condenser. And this is a picture which we can find in dark field microscopy. On the dark field we can see um, objects like with a like a balloon, like a shining object. Object is a shining. And next is a phase contrast microscopy. Okay, and in this case, we also use light microscopy, but with phase contrast optical design. Uh, Principle of the method amplifies small difference in refractive insights. When light is coming through translucent objective, we can see these changes. To change and in this process uh, we change the amplitude. We can see the changes and transparent object becomes visible. This type of microscopy can be used for observation of transparent object. Okay, this is phase contrast light part, and in the case we can see like we have here we can see usual objective and there are we can see specific phase plate which refract or diffracted lights and in this process is we can see the changes of amplitude. That's why translucent object became visible. Okay, so we can see visible object. And luminescent microscopy. In this way, we use specific luminescent microscopy when and the luminescence of the object is ultraviolet light is registered. So we use ultraviolet light, not usual white light. And application in microbiology is microscopy of specimens stained with fluorescent dyes, evaluation of serological fluorescence, and like that. And this is picture of luminescent microscopy. On the dark field, we can see colored with shining objects. And electron microscopy effect is uses beams of electrons instead of light rays. So another principle, not the same of light microscopy. And this method is used for observing the viruses, ultrastructure of microbial cell, and like that, because we can see the smallest part of bacteria. Like that, here we can see bacteria in electron microscopy. Okay. That's all. Thank you for attention. And if you have any question, please ask.